Hello and welcome. You're listening to Cellvita, Science Talks, where we explore key challenges in drug discovery and development. In today's episode, we're diving into the topic of cell-based potency assays, as discussed in one of our latest blog articles, Where Biology Meets Statistics, Solving the Potency Black Hole. In this piece, we take a closer look at the complexity of cell-based potency assays and how biology, assay design, and statistics come together to ensure that a therapy truly works as intended. The full version of this article, including all figures and graphics, is available on our website. If you're watching on YouTube, you'll also see the visuals on screen. And if you're listening on Spotify, we'll guide you through them step by step. Where Biology Meets Statistics, Solving the Potency Black Hole. Challenges and Insights from Cell-Based Potency Assay Development. Determining the biological activity of therapeutic proteins is a crucial challenge in biopharmaceutical development. Among the analytical tools available, cell-based potency bioassays remain the only approach capable of directly linking in vitro results to the drug's mechanism of action. As a primary critical quality attribute, potency ensures that a biotherapeutic consistently performs as intended throughout its life cycle, from batch release to stability and comparability studies. Unlike physicochemical or immunochemical assays, which measure structure or binding alone, a bioassay reflects the drug's functional activity in living cells. This makes it indispensable to confirm that manufacturing processes maintain the product's biological efficacy. However, this value comes at a cost. Bioassays are complex, time-consuming, and often highly variable. They demand not only technical precision, but also a deep understanding of biology, assay design, and statistical analysis. From a regulatory perspective, a bioassay is essentially a combination of cell biology and appropriate statistical tools. This very premise creates challenges in understanding how to conduct studies, forcing each scientist involved to step outside their comfort zone. On the other hand, customers often perceive a bioassay as just another analytical test necessary to assess the quality of a drug batch. Unfortunately, the issue runs much deeper. While in analytical testing, the definition of a given technique already allows for a preliminary understanding of what the method is intended to demonstrate. The term bioassay is something of a black hole. For each drug under investigation, this test will be entirely different, based not only on the molecule's structure, purity, and the composition of the final formulation, but above all on a deep understanding of the therapeutic's mechanism of action. Consequently, even for biosimilar products, the final method may require modification, for example, due to differences in excipient content. In summary, the only common feature among various bioassays is the use of living cells as a model. The very selection of an appropriate experimental model, from numerous available commercially or even through development of a dedicated cell line, becomes part of the method development process. Understanding potency and the mechanism of action, the Socratic method of finding the solutions. Potency is commonly defined as a drug's specific ability or capacity to produce a given biological effect. In the context of biologics, it represents the strength of the product in eliciting its intended response. Therefore, a valid bioassay must faithfully reflect the drug's mechanism of action. For instance, antibodies may act by ligand receptor blockade or by triggering effector functions such as antibody-dependent cellular cytotoxicity. Cytokines or growth factors stimulate cell proliferation or differentiation. Each mechanism of action dictates not only which cellular model should be used, but also which readout is the most appropriate, whether luminescence, absorbance, fluorescence, or reporter gene activation. It is crucial to select a laboratory system that accurately mimics drug action in vivo in the patient's body. The closer the assay reproduces the physiological context, the more meaningful and reliable the potency data will be. These data will also be more informative in terms of drug batch characterization, allowing reflection of its pharmacological properties. Selecting the right test system. Strategy and tactics above all. Designing a bioassay begins with a deep understanding of the therapeutic molecule, its structure, receptor interactions, and downstream signaling. Literature review and database research support the selection of potential cellular models and readouts. At this stage, analysts must evaluate which cell line expresses the target receptor and how selective the chosen model is for the drug's intended activity. They must also consider the practicality of the approach, including the cost and stability of maintaining the culture. An early assessment of advantages and limitations helps identify the most promising systems before entering the experimental phase. In many cases, several cellular models are verified in parallel, particularly when the molecule acts through more than one mechanism of action. This systematic exploration ensures that the final assay setup reflects the drug's biology and remains feasible and reproducible under laboratory conditions. Tactical design: managing and monitoring the living system. Great challenges are only given to those who can handle them. Cell-based assays inherently involve biological variability. Subtle differences in cell culture conditions, reagent quality, or even analyst technique can shift results. To manage this complexity, a tactical design approach is essential. It ensures that variability is understood, controlled, and monitored across the assay's life cycle, from development to routine use. Figure 1. The figure presents a schematic overview of a continuous process linking assay design, optimization, and performance monitoring. If you're watching this on screen, you'll see a step-by-step workflow supported by decision checkpoints. The process begins with selecting an initial test system. It is followed by a pros and cons analysis, and then by initial test system verification. At this point, a key evaluation box appears, asking, are any problems detected? Is the system suitable and reproducible? What is the labor consumption, complexity, and cost-effectiveness? Based on this assessment, the workflow continues with another round of pros and cons analysis. The process then moves into bioassay development. At this stage, critical elements are defined, including assay conditions, reference standards, data analysis strategy, and initial acceptance criteria, along with checking the method's validatability. Next comes qualification and validation, where final acceptance criteria are established, all parameters are checked against guidelines, and the method's performance is verified. The workflow then continues into method implementation and monitoring. At this stage, and also during partial redevelopment, another decision point appears on the right side of the diagram. It asks, are any problems detected? Is there a need to change the assay, acceptance criteria, or data analysis? If needed, the process moves into partial redevelopment, followed by qualification, validation, and verification. Finally, the workflow loops back to the initial test system selection. This illustrates that bioassay development is not a linear process, but a continuous, iterative cycle driven by evaluation and improvement. Optimization. Through iterative optimization, parameters such as seeding density, stimulation time, and reagent concentrations are fine-tuned. The goal is to balance biological relevance with reproducibility. Continuous performance monitoring enables early detection of assay drift, ensuring long-term consistency. Critical steps in bioassay development: detecting the undetected. Once the model system is selected, development proceeds through a structured series of steps governed by USP 1032 and ICH Q14 recommendations. Analysts begin by confirming that the assay indeed represents the drug's mechanism of action, and by defining the key operational parameters, such as cell culture conditions, plate layout, and dose-response range. Figure 2. The figure presents the most common critical steps in bioassay development. If you're looking at the visual, you'll see a structured checklist that guides the entire process, from early biological understanding to final validation readiness. It begins with gaining knowledge about the cells, followed by preparing master and working analytical cell banks. Next comes determining the cell seeding density and defining the response, including the full dose-response curve and slope ratio. The process then continues with determining doses and selecting the reference standard. Further steps include establishing assay conditions, identifying critical reagents, and determining the critical elements of the assay. Next, the assay format is defined, the plate is randomized, and a uniformity trial is performed. At this stage, any non-specific response is excluded. The focus then shifts to data, starting with determining the data analysis model and selecting the appropriate statistical model. Once this is defined, acceptance criteria are established. Finally, the method is evaluated by checking reproducibility, specificity, and overall validatability. This structured approach ensures that all critical aspects of the assay are addressed, even those that may otherwise go unnoticed. Statistics. Statistical models such as the 4-parameter logistic model or the linear parallel line model are chosen depending on the response type. Factorial design of experiments may be used early in development to evaluate robustness and understand interactions between variables. Once conditions are optimized, analysts verify reproducibility, specificity, and preliminary accuracy to confirm that the method can proceed to full validation according to USP 1033 and ICH Q2R2. From development to routine control, the broader impact of potency assays. Cell-based potency bioassays are a cornerstone for biologics development, providing critical data that link molecular design to therapeutic function. They ensure that the drug consistently exhibits its intended biological effect, supporting product comparability after manufacturing changes and during stability studies. Moreover, the process of developing a bioassay, spanning mechanism of action analysis, model selection, and rigorous validation, creates a comprehensive understanding of the product's biological behavior. This knowledge becomes invaluable for troubleshooting and continuous improvement throughout the product lifecycle. Patient impact. A potency result is not just another value on the certificate of analysis. It provides reliable and trustworthy information that the drug administered to the patient is of appropriate quality, active, and stable, thereby ensuring safety of use. In other words, the development of each potency test, and especially a bioassay, involves tremendous responsibility. At the other end of the supply chain is a suffering human being. Conclusion: the development of cell-based potency bioassays is both an art and a science. It requires precise technical execution, deep biological insight, and strategic planning. By integrating these elements, it becomes possible to design assays that truly represent the therapeutic mechanism, generate reliable data, and comply with regulatory expectations. At Cellvita, we combine our analytical expertise with a strong understanding of pharmacology and regulatory science. Our goal is simple yet demanding: to transform complex biology into measurable, reliable data that drives the success of modern biotherapeutics while taking care of the patient's well-being. We are fully aware that the processes of drug discovery and drug development are two sides of the same coin. At every stage of the project, we remain scientists while never losing our sense of responsibility toward patients. Thank you for listening to Cellvita Science Talks. If you'd like to explore this topic further, we invite you to visit our website and read the full blog article, including all figures and additional insights. And if you're interested in more content like this, stay with us for future episodes, where we continue to explore the science behind modern drug discovery and analytical development.